And today we invited Dr. Navaneeth to share some information and knowledge about shrimp farming, EHP, about transformation and EHP identification and many control measures about EHP. And he have many years focus on probiotics and intensive farming practice in biofloc farming, and have many years of experience in hatcheries. So we are very honored to invite him to share knowledge with us. I hope we can listen carefully and learn more knowledge. So if you have any questions and problems with EHP, you can leave the message in the message box. And after the presentation, Dr. Navaneeth will answer you. So let's welcome Navaneeth.
Yeah, hello. Good evening all. Shall we start, Jackie? Hello. Good evening to all. First of all, I would like to thank
Hello, Navaneeth. Cannot hear you.
Now, okay?
Okay.
Yeah. Yeah, but basically the presentation includes general information of EHP, disease distribution of EHP, transmission, diagnosis, preventive measures, control measures, and summary. Yeah, enterocytosis and hepatopenia. The mic is coming under microsporidian parasites. So microsporia are obligate intracellular spore-forming endoparasite, which infect both terrestrial and aquatic host. There were around 187 genera of microsporidia, which consist of 1,300 species. Out of these species, 25 are known to infect crustaceans. The life cycle of these microsporidian parasites are very complex. 3 genera of microsporidia are known to be reported in pinnate shrimp is infective. EHP is the emerging microsporidian parasite which infect pinnate shrimp. As the name indicates, EHP is infecting shrimp hepatopancreas. The spore is made up of thick chitin ball, which helps the spore to survive in harsh environment, and size of the spore is around 1 micron size. EHP is first detected as an unnamed microsporidian from growth retarded black tiger shrimp from Thailand in 2004. And there were other reports of resembled unnamed microsporidians in hepatopancreas of Pinus monodon in Malaysia, China, and in Pinus japonicus in Australia 2001. EHP is first characterized and detailed and named as new species in 2009 from Thailand. Now EHP is reported in all of the most shrimp aquaculture active areas. In India, both coastal states as well as in inland states, EHP is reported. This is the generalized symptoms of EHP infection in shrimp. At the starting stage, the appearance of white gut or white fecal will be occurred. In this stage, hepatopancreas is severely damaged and the cells were getting sloughed off from the tubules of HP which containing high amount of reserved fat cells and HP cells. It is coming through intestine, and the damaged sloughed content uh, appears to be white and float in pond water, which we called as white fecal or white gut. After diminishing the white fecal from the animal moves to loose cell because of the improper digestion and absorption of the nutrients by the damaged hepatopancreas. As the hepatopancreas become regenerative, the shrimp may recover from loose cell, but during the time, permanent stunting or growth retardation will occur that leads to size variation. After this disease occurring phase, there will be a chance of getting secondary bacterial infection, which leads to mortality. And the cycle continues in every 35 days, every 35 to 40 days. Due to the secondary damage, the mortality rate will be 5 to 20% or even more, depending on the pond condition. The surveillance from our study shows that in India, in 2013, very limited case of EHP incidents were reported. When the time goes, the first appearance of white fecal or white gut during the culture period is decreasing every year. In 2014, the first appearance of white fecal is to be reported in 50 to 70 DOC, whereas in 2017, it's reduced to even 25 to 23 DOC. That means the number of EHP spore is increasing and accumulating in the pond during the culture. In our studies, the main clinical symptoms of EHP is color change and shrinkage of hepatopancreas, white gut, empty gut or watery gut, and loose cell. In our study and field observation, all the white fecal is tested positive for EHP, and all EHP positive ponds were observed to have white fecal occurrence during the culture period. Pictures of EHP infected, both experimentally and naturally infected EHP infected shrimps. We can see clear shrinkage of hepatopancreas, discoloration of hepatopancreas, and white gut. See the color. This is the natural healthy EHP, natural color of hepatopancreas of pinnate vannamei, whereas in EHP infected shrimp, the infected shrimp hepatopancreas become very pale and it is shrink in size. This is the picture shows the loose cell severity of the EHP. Even though animal is in live, it is loose cell is so much severe, which leads to secondary infection and mortality in shrimps. This is the experimentally infected shrimp showing different fecal coloration. First one is EHP infected without no color, and second B, this one shows the intermediate infection. That means early stage of infection and the clear white fecal. This is a comparison of normal and naturally infected naturally infected EHP vannamei. So you can see in the first picture, the hepatopancreas, outer white color is the epithelial lines which we observe through shrimp. Inside, thick red or pinkish color is we can observe. They indicate the healthy shrimp hepatopancreas. Whereas in EHP infected hepatopancreas, sometimes in this picture the epithelial cells, but inside the content. This is the very much important. The cell is getting fat deposition is sloughed off, and the cells also sloughing off from the hepatopancreas. This is naturally infected from pond from Iskapalli, Andhra Pradesh. This picture shows the starting of white gut, because it is not in complete gut, it's starting there, but you can see the discoloration of hepatopancreas. So in field level, when we are observing when we are observing the animals for EHP, you can see in this portion where the intestine as well as the hepatopancreas meets. So if you find any discoloration, not in the outer layer, inside, that may be the indicator. You have to see there. If there any white coloration or any gap or some white fecal floating or watery gut, we will call as watery gut is present, means that is the chance of possibility of EHP infection. Once the EHP infection starts, the gut become empty, watery, then all the reserve fat cells, almost it will become sloughed off. So that's why the white color is forming. So transmission of EHP in farms and hatchery. In farm, the source of EHP is mainly from post larvae, because if in a new, there are some reports in inland as well as in new farms, there are reports of EHP incidents. So that means the major source of EHP in a farm is post larvae, or sometimes the existing spores from previous culture, or the contaminated water from source. Sometimes farmers may not be doing proper chlorination or water treatment. So where if the water is from canal or creek water, if the creek is contaminated with the EHP spores, the same spore is getting to our pond and it is getting contaminated our pond. There is a chance of in that case also there is a chance of getting EHP infection in the pond, especially in biofloc ponds or those who are doing HTP line. That will be the two reasons. There will be the two reasons for EHP infection. One is predominantly from post larvae or from our pond water or from source water. In hatchery, the main contamination source is live feeds like polychaetes, Artemia biomass, oyster. So polychaetes is a natural carrier for EHP, because it is a detrivorous organisms. It will eat all the materials, all the waste materials from the surroundings. So once the creek is contaminated with the EHP, the polychaete also getting a chance to have have EHP spore in the body. But polychaete is not to be a host for EHP. Same way, Artemia biomass also. There are so many reports from China, Thailand, and even India also. Artemia biomass is prone to be a host for EHP. Second thing, water. The water contamination is very much important in hatchery field also, because from the source water, because all the hatcheries are in the coastal lines. So there, the end water, the coastal line also getting, there's a chance of getting contamination with the EHP spores. Then the broodstock quality. Sometimes pond bred broodstock, or earlier in quarantine facility also, EHP was not screened, it's not mandatory. So there is a chance of getting contamination from that area also. Now what is happening in India is the cycle is continuing. Hatchery to farm, farm to natural water bodies to live feeds, then again hatchery. The cycle is continuing. In experimental of EHP, the transmission assays, the feeding experiment, cohabitation, soil sediments, pond water, all these routes are animal is getting EHP. The bioassay reveals that EHP can be transmitted through feeding, cohabitation, soil sediments, and pond water. In feeding experiments, on 5th day of post-infection, 100% of animals were getting positive for EHP. That means in pond condition, the cannibalism plays severe role in super spreading of EHP in the pond. Whereas in pond water, 100% of EHP infection only occur after 30 days of post-infection. That means through water, EHP spread is occurred, but in a very slow manner. Very slowly only it is spreading. The same way, cohabitation in the sense when EHP infected animal is put together with the EHP negative animal, through the cohabitation is spreading. On 5th day onwards, EHP is getting positive, but on 15% of EHP infection is occurred. The same way, soil sediment. So these experiment results suggest that better treatment of soil, better treatment of pond, and low stocking. If the pond is having higher stocking density, the cohabitation chance, here we can see cohabitation chance of getting EHP infected is getting more. So better to have a low stocking density to control EHP. The same way, there are other reports of EHP infection in peanut species. That means P. vannamei, Monodon, Japonicus, Indicus, Metopenis dobsoni, all are susceptible to EHP. And there are some reports like mud crabs, Artemia, polychaetes are positive for EHP. But still we have to find out whether it is a host or carrier. Being a crustacean, the mud crab may be the host of EHP infection, but there is no conclusive evidence. But in case of polychaetes, it's a purely host. It's not a pure host, but it's a carrier for EHP. In the same case, Artemia also. This is a published paper conducted in India, where the polychaetes, the Morphasia, what we called normally called as mudworm, to carry out to screen to find out EHP, whether it is a carrier for EHP and other pathogens. So almost 447 individual samples were collected in 3 different states: Kerala, Tamil Nadu, and Andhra Pradesh. In this study, almost 7.2% samples were positive for EHP, whereas WSSP is little bit positive in 5.2%, and IHN is completely negative, and mixed infection only 2.2%. In this result, in wild area, we are not getting any WSA infection and very less contamination with EHP in wild area. So 21% of polychaetes collected from farming area were positive for EHP, and 28.8% were positive for WSP. 12.3% of polychaetes were positive for EHP and WSP in culture area. So that means the culture area, the water from culture area is contaminating the natural bodies, which is contaminating the polychaetes and other invertebrates, which is a carrier for, or act as a host in different organization level. When the polychaete is taking as a live feed hatchery, the cycle is continuing from hatchery to farm, again farm to hatchery. So in our study, in conclusion is that EHP is not a natural parasite of polychaete worms. The increased prevalence of EHP is 25% and WSV is 28%, and severity near only in shrimp farming area. EHP is likely to spread from shrimp farming sector. A potential risk to broodstock in hatcheries through live feeds. 4% of mixed infection, both WSP and EHP. So these are experimental studies on transmission of EHP from infected shrimp to polychaeta and vice versa. The studies included both life cycle screening of life cycle of polychaetes which have infected with the shrimp, which is infected with EHP, and it shows that there is no evidence of vertical transmission. That means it is not a host for EHP is not a host for polychaetes, but it's a carrier. Coming to detection of EHP, there are different staining methods can be used like Giemsa stain, H&E stain, hemoxylin, Weber modified trichrome stain, Ryan blue trichrome stain, floxin B, and some specialized staining methods like calcifer white staining, cytox stain, propidium iodide, and calcifer white stain to identify the viability of the spores, and electron microscopy of TEM and SEM. So for this clear image, for a clear identification, we have to isolate the spores in a purified way, because the spore size is 1 micron and it mostly coming on the size range of bacteria and yeast. So in order to purify the EHP spore, the Percol gradient chemical which is used for gradient preparation is Percol. The Percol gradient which allows the spore separation from debris. Through this method, we can separate the spore in a purified form to conduct other studies. So these are the different microscopic pictures of EHP spores. First one is fresh microsporidian EHP spore, which give an olive green color, in an oval shaped olive green color. We can see in 100x. We can barely see in 40x, but clearly we can see in oil observation because of the size. The second one is Giemsa stain, third one is trichrome stain, and fourth one is floxin stain. So all this staining method, direct microscopic observation as well as floxin B stain is very reliably easy method. So in farm level also, if they have the facility, those who are having the facility of microscope easily can stain with floxin and observe the EHP spores. So this is the one specialized staining technique. It's a fluorescent microscopic technique. The stain used is calcifer white. You can see the blue stars like spores in the HP tubules. These are the tubules. You can see the magnitude of EHP infection in the hepatopancreas. So these are the sections of EHP infected hepatopancreas. So first one is H&E staining. You can see the clear distorted HP tubules and cells. First one is H&E stain. Second one is semi-thin section of EM. You can see the blue dots that is heavy infected in almost all the cells. Done one is the trichome staining of the thin semi-thin section, which is heavily infected my EHP infected hepatopancreas cells. You can see in different life stages of EHP in the same tissues of my hepatopancreas. So this is the picture. This is a transmission electron microscopy of healthy and infected EHP infected hepatopancreas. The first picture shows healthy microvilli and the tubular space. Microvilli absorb all the nutrients. Second one is HP cells completely damaged and the microvilli structure is completely distorted. So in EHP infection time, all the microvilli is getting sloughed off. Eventually, the cells also getting sloughed. The microvilli are the main important structure for absorption of nutrients. So once EHP is infected, all the basic absorption structure is getting subflow. The absorption is not happening, and animal is getting loose cell or white gut. So this is one of the interesting picture. You can see in one cell there are matured spores, whereas nearby another reinfection is happening. That means even body is infected with EHP and it is going to slough off, or it is going to produce mature spores, again reinfection is happening. So the severity is much more higher. The second picture is the term image of EHP spores. It's in an oval shape. You can see the polar tubes. Polar tubes is the syringe-like structure which helps the spores to introduce a sporoplasm into the cells. Yeah, this is the EHP infection stage. You can see the spores size of 1.15 micrometer size, and you see the length of the polar tube. It's almost 28.35 micrometer. So this much big polar tube structure. The polar tube structure is this much big. And the second stage, the first picture, you can see some red color sporoplasm or green color sporoplasm is ready to inject in the cells. So to control EHP, there are two options. One is, before infecting the cells, it has to be inactivated, or within the cells. So better method is, before infection it has to be inactivated. So the use only biosecured live feeds like polychaetes, Artemia, oyster. Every live feeds has to be checked and ensure the absence of EHP. The best and available method to ensure EHP-free seed, EHP-free live feeds is make them frozen. So some, even the quality of the output from the hatchery may be reduced, but it is ensuring the frozen live feeds ensuring complete biosecurity from EHP. Because EHP is inactivated in different temperature like 4 degree or -20 or -80, and -80 or quick freezing is the best reliable method to inactivate the EHP spores, especially from polychaetes, oyster, and Artemia biomass. And ensure the SPF broodstock status frequently. That means even though after quarantine the broodstock is certified as SPF, every hatchery, every maturation unit has to check its biosecurity status or SPF status every now and then. In a frequent intervals, it has to be checked. Even though, unlike other testing method, EHP testing is a non-lethal method. We can use fecal threads from the maturation section to track down the EHP status and maintain the biosecurity in hatchery in all aspects, and in developing infrastructure. It's already known that water also contaminating, and through water contamination, EHP is entering in the system. So we have to increase the infrastructure like water filtration, UV filtration system, ultra filtration system, like that. It will give much more advantage to get rid of EHP from the system. And hatchery people has to be focused on producing live feeds like polychaetes or other things to ensure the biosecurity. So this is a typical example of SPF polychaete culture facility in Saptagiri hatcheries. Only through SPF polychaete and live feeds only we can achieve the SPF EHP free status PLs. These are the most advanced water filtration system and UV filtration, UV disinfection system in the hatchery. In farms, okay, is free by the PL should be tested for tank. Ceremonial method and fecal threads. Number identify EHP, and moreover make use of advantage of a nursery system, because in farm level nursery system is very much important, not only for growth purpose but also from the EHP. Why? Because nursery system is growing in a small confined environment. It's like a sheeted ponds or earthen or above ground ponds. Once we are growing for 30 days, 25 to 30 days, the PL is getting PL. If EHP positive is there, means within 25 to 30 days we can do the screening and we can find out whether it is EHP positive or not. Because in a cohabitation scenario, EHP is getting infected very fastly. So as in the experiment itself, once the PL or a shrimp is infected or ingested with an EHP spore, it will take minimum 5 days to expel the spore to environment. That means it needs 5 days. So in PL testing, there is a limited chance. If the PL is heavily infected or severely infected from the broodstock or from the napoli, the chance of getting PL positive is high. But it is even very low, very less infection is happening in the PL stage or in the napoli stage. Getting positive for PL is very, very less. So in nursery system, it is almost 99% become positive for EHP if the PL is infected in earlier stage, which cannot be detected from the at the time of stocking. And after that, all the farm equipments like aerator, blowers, aero tubes, or in paddle wheel aerators should be disinfected properly with the proper disinfectants. This is one of the experiment. It's like before in a farm, the soil treatment was done with the sodium hydroxide at 200 to 300 ppm. It's like a spraying. So soil hydroxide solution, so the sodium hydroxide solution at the concentration of 200 to 300 ppm made, and it is sprayed on the all over the surface. After 6 to 12 hours, acidified chlorine, that's 200 ppm hydrochloric acid and 200 ppm chlorine, mix it together uh, and spread over to bring down the pH to normal level. After drying, the pH is normalized and soil test of the pH EHP has been done. At that time, it is negative for EHP, and the pond can be ready for next stocking. But the problem with hydroxide solution is it will affect the fertility of the soil. So sodium hydroxide is not a regular treatment solution for EHP. So again, one proposed pond preparation method there after EHP infection. Disinfect the water with chlorine or other commercial products. And it is not that cheminophore, potassium permanganate, is very good disinfectant, or it's a very good very good chemicals for inactivating EHP spore. At 15 ppm for 15 minutes, complete EHP inactivation is happening. So if the pond is unfortunately got infected with EHP, after harvesting, keep 1 feet water of pond and apply 12 to 15 kg of caminophor, depend upon the organic load of the water. So once if it is treated with caminophor, you can drain the water after discoloration. So it's very much important that to see the discoloration time of water, because when the pond is having more organic load, the discoloration will be coming fast. So there will not be enough concentration or enough time to inactivate. There will not be enough concentration or enough time to deactivate or kill the spores. So it is very much important to see the discoloration. So after discoloration, dry the pond for at least 15 days, then raise the soil pH by 12 to 12 by calcium hydroxide or quicklime or burn lime, or you can use sodium hydroxide. That also you can monitor the rising of the soil pH. Depend upon the soil capacity, soil buffering capacity, the pH may be raised very slowly or very fast. So depend upon the soil type, you can fix the dosage also. Say example, 2 to 3 tons, or some place it may be goes up to 4 to 5 tons per acre. Generally, 6 tons per hectare is 6 tons per hectare is the normal dosage. And once the pH is increased, most of the EHP spores can be inactivated. But here also one application process is there. Initially, you can add half tonnage of lime to the pond and plow it crisscross direction, so that the pond soil mixing will be proper and all the lime content, it will be penetrate into the soil. So it will be, there is a much more chance to inactivate all the EHP spores which present in the ponds. And these are some of the photographs of potassium permanganate treatment in the ponds. So this is one of the schematic representation of disease progress from hatchery to grow out. So in hatchery, very less chance of getting EHP. So very less. When it is coming to the nursery, it is the magnitude is increasing. When it is grow out, it is become billions. So there are two entry points where we can stop the EHP. So first one is better diagnosis in the hatchery level while taking the PL. If the PL is passed, again you can do nursery. There also there is a chance of getting EHP treatment in the EHP testing in the nursery itself. Once if it is passed, then you can go for the grow out. That's why we can save our crop as well as in the culture. EHP spore number is increasing in and accumulating in the pond environment. So Enterocytosomen hepatopinae is recently introduced pathogens that has become one of the major sanitary risks in Indian shrimp industry. EHP is the problem that needs an urgent control. Geographical spreads in India is mainly through seeds, aquaculture inputs, especially in inland areas. The strict adoption of BMP for broodstock, hatchery, and farms is to be taken. So, if you have any doubts, I think it's better to have a little more interactive session. Then we can have some chat on you can clarify some doubts also. If you have any question, please ask.
Thank you very much. Very useful information, and share the information about the EHP introduced to us from many aspects. So if you have any questions, please ask. And
yeah,
hand up. Also can hand up.
Yeah, EHP. Yeah, one question from Tirupati. EHP, how much of depth of present in soil? Yeah, how much depth EHP can go in the farm soil? So it's depend upon the pond condition, and we can say, if the pond is having some dollars, borrows like polychaetes or something, it will be go very deep down, because polychaetes also having also will take EHP spores, and it will go into the burrows, it will deposit the fecal matter there. So it depend upon the condition, and it depend on the pond bottom. Mostly when we are doing lime treatment, almost 2 to 3 inches EHP in our experiment also 2 to 3 inches we are found EHP spores actively, and in our experiment also like that soil only be collected, and it is infective. If the seed is free from EHP, any chance of getting during pen farm? Yeah, of course, EHP, if the seed, first of all, if the complete EHP is completely free from PL is completely from EHP, if you are getting any positive in during farming means first thing it's a cross contamination from or pre-existing spore from the soil. So there are a lot of chances are there. There are a lot of reports are there. Even though PL is completely from EHP, there is an incidence to getting EHP in growing culture. So that is from soil as well as from water.
Okay, any other questions?
How to treat EHP from EHP when shrimp is infected? Yeah, actually there is no treatment for EHP infection in shrimp, but you can have a preventive measures like giving good organic acids, good bile acids, and to reduce the infectivity as well as to increase the metabolism or to increase the digestion and absorption of shrimp, which help and protect the hepatopancreas, so that the growth won't be retarded or growth cannot be compromised. And of course you have to use good probiotics to protect the shrimp from secondary infection of other bacteria like vibriosis. Agriculture found not EHP positive. In a nursery culture, farmer noted AHP positive. What he can do? It's better to abandon culture to dispose the EHP seed of the pond in the nursery itself, because if EHP positive means the nursery only is get contaminated. So it won't, if you are shifting the seed to pond, the entire farm area will be contaminated. So it's better to abandon the seed, kill the seed, dispose the seed, then do the new culture. And before stocking again in the nursery, it's better to clean all the things, decontaminate all the things from EHP. Question nursery, is it need to drop culture, or yeah, it's advisable. It's better to drop the culture. once EHP problem occur in hatchery, is it continue to run hatchery? So first of all, you have to find out the root cause. In hatchery, you have to find out the root cause, whether it is coming from maturation section or if it is coming from NRC. If the hatchery is NRC, Napoli Rearing Center, means you have to find only two chances there. It is from Napoli or it is from this one or waters. So you have to find out the root cause. Is there any method to cure EHP nursery product? Curing EHP is very difficult because we are also running the experiments. That means in 120 days also EHP is not get cured because it's still in the body, but animal is growing, and there are some farmers with EHP infection with good control of good products like eukaryotics and all. People are taking harvest in 50 counts, 20 counts, 30 counts like that. So even in the EHP infection time also. So it's not a pure, we have to help the animal to grow safer even though with EHP infection. Is there any bacterial strains to control EHP? EHP is like a intracellular parasites. So if there is no bacterial strains to control EHP, like it's not, there is no bacterial strains to control EHP. The only thing is that in the EHP infection time, you have to use some of the good products like bile acids, that and all helps and protect the shrimp, protect the shrimp hepatopancreas, which is helping better digestion and better absorption.
Okay, very thanks. Very useful information. And now we also invite our Dr. Bain to share some information and products to control EHP and Vibrio. So let's welcome Dr. Bain.
Okay, can you hear me, Jackie?
Yeah, yes.
Hello, everybody. First, I'd like to say thanks to Dr. Navaneeth. Thanks for your very useful information from the pond preparation, how to prevent, how to treat the EHP in the shrimp farming, as we all know, especially in the late part of 2021. There are plenty cases which infected by EHP and cause a huge loss for the farmers, you know, from the Indian, China, Indian, Vietnam, Thailand, and Indonesia, all the main producer for the vannamei shrimp. Was that is very bad for the farmers. So what I like to share today, because in China from the last year we have totally banned the antibiotics for the shrimp farming. So and sometimes if the shrimp infected by the vibrios or the harmful bacteria overloading will cause the shrimp stress. Then finally maybe the EHP will easily infect our shrimp and make the shrimp farming much difficult. Okay, so what from the last year, we are the whole industry, the shrimp farming, as you know, we are the biggest producer in the world. Of course, we are also the biggest consumer in the world. Every year we import around 1 million tons from overseas, from Ecuador, India, Vietnam. So we want to find out how to control. I mentioned only control or prevent, not treat the EHP and also the vibrio problem. You know, once you got such kind of problem, maybe your culture will difficult. Make your culture difficult. Okay, from last year, we are trying to use some, the animal health product to improve. Also, just now, Dr. Navaneeth also mentioned that we have to improve the immunity of the shrimp. This is very important. When the shrimp health, okay, so the pathogens won't affect easily. So from last year, we use what? We use the Chinese herb, we use the essential oil, we use also some probiotics to try to improve the immunity of the shrimps. And we did get the positive result. Let's say for the EHP, because EHP is, EHP is intracellular, that means live in the cells, not easy to kill. But at a certain stage, when they release out of the cells, we're trying to use some essential oil to kill the spots. This is possible, but for the spots live in the cells is not easy. And we use the, eucalyptus essential oil from Australia, and this is really got a positive result. After using 1 or 2 weeks, the spots, the number of the spots reduced significantly. This is, of course, this year we continue to trying all over the world. Okay, last year is the first year, and this year I think we will get a big result for the farmers. This is hopefully we can help the farmers to solve the EHP problem. If you like, you can try to use some essential oil, you try first. Okay, but this is only for the prevention, not for fully kill the EHP and get rid of all the spots from the shrimp. That is impossible, but we can reduce the number of the spots in the shrimp itself. Okay, this is one part. Another, I think maybe we also facing the what problem, vibrio problem. Parahemolyticus is the most the number one disaster for the shrimp farming in the last year for China. Okay, so what we do, we use some organic acid plus some essential oil will work very well. And today I got the report from industry. I think Jackie, I will let Jackie to translate and publish, so you guys can have a look and can try, see whether get the satisfied result. For our Calford, is purely is above 98% and more easily to absorb, and our calcium ion is above 30.1%, and the absorption rate is about 90%, which is much higher than the the other commercial products. Okay, so once you got the vibrio problem, you can try to use the Calford plus essential oil. After 3 days, you will see good result. Jackie, you can move to next slide, which is agar plate. Agar plate. Next one. Next one. So for the component, I'm not going to explain because this is a very, very scientific and common in the market. You can search, and you also can get from Jackie. Next one. Next one, the latest one. Next one for the vibrios. Okay, the back. Okay, this is I just got it because now you can see I'm in the station, the high-speed train station, just back from shrimp from the farm. Okay, we did this trial for 3 days. Okay, you know we use the careful 5 to 8 gram per kilo plus 2 to 3 gram per kilo eukaryotics. So after 3 days, the left side, okay, you can see the Vibrio is very less, but the right side is in the water. We test two. First, we test the Vibrio in the water, and the second is for the Vibrio in the shrimp. After 3 days, we check again. So the Vibrio in the shrimp is very less, but in the water still got some number. But the good news is even the water have, if you have some pathogens or vibrios, if your shrimp not under stress and healthy, definitely they won't get some problem. Okay, this is a good news. So first we have to treat the vibrio in the body, in the shrimp body. Okay, so this is a very how to say mature and very effective. This is what I like to share to everybody. I think also I like to invite Dr. Navaneeth to share some method to treat the vibrio or share some information from Indian industry. Jackie, I think that's all from my part. Hey, Jackie.
Okay.
Yeah.
Thanks a lot, Dr. Bing.
I think I'd like to invite Dr. Navaneeth to share some knowledge or information on vibrio in Indian shrimp farming industry. So how to treat the vibrios in Indian? Okay. Hello. So you just ask the doctor to.
Hello.
Hey, doctor.
Hello.
Hello. I can hear you. I can hear you.
Hello.
Just go ahead. I can hear you, doctor. Okay.
Hello, Dr. Navaneeth. We can hear
Yeah.
you.
Hello. Am I audible now? Hello?
Yeah, I can hear you.
Yeah.
I can hear you. Yeah.
Yes. Actually, some network issue is there, I think. Sir, regarding Mr. Arunjit, it's one question from Arunjit. Is it possible the treatment process in Kerala? Yeah, you can try KMNO for at 15 ppm level. So because Kerala, we cannot dry the pond completely, no? So you can try KMNO for treatment at 15 ppm.
Yes.
And it works well. And
Thanks.
one more.
Yeah.
Okay,
So
thanks a lot.
Yeah.
Any question about the EHP? And you can ask and leave the message. Or any question about the product comfort and eukaryotics. Any questions? Okay, if there no questions, thanks for your time and thanks for your participate, and thanks for Dr. Navaneeth and Dr. Baines sharing. So if you are interested about our webinar and meeting, you can join at our WhatsApp group, and we will share more information about shrimp farming later and hold many activities.
Okay.
Okay,
Thank you.
thanks, Navaneeth. And
Thank
also you can add my WhatsApp. Thanks, everyone. Thanks for your time. Okay, bye-bye. Have a good night. Yes.